Me too. All right, recording. Okay, I am the seminar host for the day. I am Lauren Esposito, so if you need anything, you can chat at me or holler at me, I guess. I am really excited today to introduce our citizen science team. We actually have the whole team, I think the whole team, is here to present about all things citizen science, and they're gonna introduce themselves. They have a slide so that you can see who they are before they switch the screen over to the slides, or after they switch the screen over to the slides, because you won't be able to see their faces for the rest of the time that they're talking. Just a reminder, everybody, please mute your mics and turn off your video if you are not needing to be seen, because it will help with the bandwidth for the seminar. I also just wanted to give you some rules and guidelines about questions. We're going to take questions from everybody in the audience at the end of the talk. And I'm gonna moderate those questions. So if you have a question for any of our speakers, just go ahead and in the chat write that you have a question. You don't need to write the question, just put a question mark or write a Q, and I'll make sure that I call on you when we have time for question and answers at the end of their talk. So without further ado, the Academy Citizen Science team.
And Tony.
and Tony from iNat, I'm so sorry. And Tony from iNat, who does citizen science in his own right, just not directly as part of that same team.
All right, I'm going to share my screen here so you guys can see our talk. And all right, presenting. There we go. And present. Everyone see that? Yes. Awesome. Good. Well, hi, everybody. Thanks so much for coming to our virtual seminar today. We are going to be talking about citizen science now, which was pretty much the only title we could come up with when asked for a title for this talk. We're just like, I guess we'll just talk about what we're doing right now, and kind of how COVID-19 is changing or not changing what we're doing at this present moment. So just to really quickly show you who's talking today, we do have the full citizen science team. So myself, Allison, and Rebecca, who's the co-director of the citizen science department, Gio and Annie, who work with us on specific grant-funded projects, and then Tony is here from iNat as well. And the overview of the talk is basically going to be, we're going to give you kind of a high-level look at what we're currently doing in the citizen science department, the current work that's going on. We'll do a little bit of a focus on the City Nature Challenge, just because that is happening in less than a month and it is still happening, but obviously will look a little different this year. Tony will talk about things you can do on iNaturalist, especially things you can do at home besides just making observations. So he's got some cool things you can do to help out other than just making observations. We will highlight some other citizen science projects that you could do from home. And then we'll end with a little focus on citizen science projects and some cool open innovation work that's happening around COVID-19 right now as well. And I will say that we have a lot of links and stuff in this slideshow that you'll see pop up, but we have a resource. We've compiled them all in one place, and we'll share the link to that at the end of the talk, and we can paste it in the chat as well. So if you do see something that sounds really interesting to you and you don't have a chance to write down the link to it, that's fine. We do have a resource that has them all together after that. So I'm going to start just with an overview of our citizen science program. The general goals of the work that we do in citizen science are about, one, connecting people to their local nature. We really like to focus on the fact that biodiversity is everywhere. No matter where you are, there is biodiversity around you. It's really important to us to build community around nature, and we do that in person through a lot of our work, but also online through having iNaturalist as our platform. We really like connecting people with other folks who are interested in nature, interested in learning more about nature. And then we also have a big goal, clearly, of collecting biodiversity data that can be used for science and management and conservation. And specifically around that goal, we do a lot of work around helping people to understand the science that happens at natural history museums is kind of like a sub-goal of all the work that we do and why we do the work that we do, because we really want people to understand what is the science that happens at the Academy. So when we look at it, like at a specimen that we have at the Academy, a specimen has a location, where it was found, it has the date it was found, we know who collected it, and then we have the evidence of the organism itself, which is the specimen itself. And one of the main reasons we chose iNaturalist and started working on iNaturalist as our chosen platform, even before they were part of the Academy, is that that exact same information is captured in an iNaturalist observation, that we have a location, we have a date, we know who the observer was, but instead of an actual physical specimen, because we didn't want people all over the world mailing us their specimens, we have the evidence as a photograph, or it can be a sound on iNaturalist as well. And both our database specimens and research-grade iNaturalist observations go to GBIF, the Global Biodiversity Information Facility. If you're not familiar with it, it's basically a giant database of species occurrence records. And so much of the work that we do when we're out there, doing bioblitzes or running programs is that we constantly talk about how these species occurrence records, how the data in aggregate are used to understand species distributions, changing ranges, and things like that. And we really emphasize that every observation is important, whether it's going to be used right now or it might be used 10, 20, 30 years from now, but every observation is important. One of the other parts of the work that we do, beyond the fact that our data go to GBIF and can be used for those answering kind of the big scientific questions, is that we almost always partner with a management organization in our work, so that those data can also be used more immediately to help our management partners make informed decisions about the land and the species that they manage as well. And I'll turn it over to Rebecca to talk more specifically about some of these programs and how we do that work.
Thanks, Allison. So Allison mentioned bioblitzes, and if we were in person, this is where I would ask everyone to raise their hand if they've been at a bioblitz, but I can't actually do that or see you. But I'm assuming many of you have been to bioblitzes or have heard us talk about them or know about them. So bioblitzes are when we get a bunch of people together in one place to work together to document as many species as possible. And as Allison mentioned, we almost always do this work in partnership with local land managers, so those data are used immediately or can be used immediately, not just as a collection of species occurrence records on iNaturalist or GBIF. So we bring people together. So next slide, Allison. Somebody's taking over presenting. I wonder if we can have George stop sharing your screen and recording whatever's happening right now.
Hi, if your name is George Bell, can you please stop your presentation so that we can resume ours?
Are you able to just reload yours, Allison?
Okay, good. Okay, so when we meet at a bioblitz, we all come together and gather and talk about what we're doing and why we're doing this work. And the main goal at a bioblitz is, like I said, to document as many species as possible. And even though a lot of people that attend bioblitzes have specific taxonomic expertise, that they are not necessarily pointing out everything they see. They are working together with their team. They're modeling good naturalist skills. Of course, they can talk about all the different things that they see together and share that expertise, but really it is everyone being curious and discovering together. So next slide, Allison. So once we meet and get oriented, we all divide up and explore different parts of the land or the park that we're working in. And so instead of having taxonomic groups, like we don't have a bird group or a plant group, we divide the park into areas so people can look for everything in those different areas. We've found through doing this for a long time that by kind of not telling people exactly what they're supposed to be looking for, like not telling people you must be looking for birds, but allowing them to be curious and discover on their own, we document a lot more together. Next slide. So you can see this is just an example from a BioBlitz at Huddart Park last year during the City Nature Challenge. And this is a map of just all the observations that we made that day. Next slide. And for those of you who don't know, iNaturalist will, when you set up a BioBlitz project, will automatically aggregate all of the observations that are made. And you can see here is what we did together. 30 people uploaded their observations, even though there were many more people there. A lot of people share accounts. And together we made 854 observations of 185 species. Next slide, or next. iNaturalist automatically makes a leaderboard for you, so you can see who made the most observations, who saw the most species, and what was the most, or what were the most observed species. And like Allison said, we encourage people to make observations of everything. Every observation is important. You know, even the 42 observations of tan oaks are important and can be used for management and also for science. And if people are curious and want to know about that tan oak or want to know what it is, it's important for them to be empowered to take pictures of everything they possibly can. Next slide. But sometimes you make discoveries and make observations that are not super common. And so this is a little woodlouse. So you imagine at BioBlitz we do a lot of turning over logs and putting them back respectfully. But there's this picture of a woodlouse. You can see it's like not a fabulous photo, right? It's dark and light, but this photo was taken by one of our friends and colleagues, Jennifer Reisinga, and she uploaded this to iNaturalist. Next. And for those of you who don't know, Scott Laurie is actually an expert, or amateur expert, or expert on wood lice, and saw this observation and thought it was something really interesting. Based on the morphology, he ID'd it as this species that hadn't been seen in the Bay Area since it was described in the 30s. And so this was a rediscovery that was made during, next slide, Allison, that was made during our BioBlitz. And you can imagine when you turn over logs, you see lots of really common introduced wood lice. And it's important though to take pictures of everything you see, because you never know if you're gonna take a picture of a invasive wood louse from Europe, or if you're gonna find something, make a rediscovery of a species that hasn't been seen in a really long time. Next slide. So we've been doing BioBlitzes across the Bay Area since about 2013, and we have held 113 BioBlitzes. This is a map of all the observations. So together at all these BioBlitzes, we've made over 63,000 observations and worked with almost 2,000 people. And we've learned a lot through this work about the biodiversity of the Bay Area, and we've built up a really nice community of folks that help us do this work. And I'll talk a little bit more about how we've used this BioBlitz as a building block for more work, but I want to turn it over first to Annie to talk about other things that we've learned through citizen science work.
All right, so I'm Annie. I work on a project in the citizen science team called Learn, Sit, Sci, where I'm investigating how and what youth learn when they participate in citizen science programs that are led by natural history museums. This project is funded by the NSF in the US and then the Wellcome Trust in the UK, and we have a bunch of partners. But what we're really looking at, next slide, Allison. So we're basically trying to answer these three questions about what youth do when they participate in citizen science programs. So first of all, what do they do? What do they actually participate in? There's been a lot of research around what adults do and what young people participate in kind of in the environmental education world at large, but not specifically what they participate in citizen science programs. And then to what extent do they develop an understanding of the science content? To what extent do they identify roles for themselves? And then we're also investigating to what extent they develop a sense of agency, which is something our PI kind of coined, environmental science agency. And then which aspects of those natural history museum programs help foster those outcomes. So how can we inform the field to make sure that youth are getting as much out of these citizen science programs as possible? Next slide. So to answer these questions, we have a big team of researchers. We have 3 natural history museums. So here at Cass, and then the natural history museums in LA and London. And then we have 3 educational research institutions at Davis, Oxford, and the Open University in London. And this is a 4-year project. We're currently about midway through our third year. We'll see how it gets affected with all the COVID stuff, but hopefully we'll get to our fourth year next year when we can do all the writing and dissemination. So we're really just starting to get into analysis, and I'll share some preliminary findings, but it's kind of just a teaser, so stay tuned for more later. Next slide. So at each museum, we're studying 3 different settings. We're studying short-term citizen science programs, which are bioblitzes for all 3 museums, even though they are very different throughout the 3 museums. Ours look a lot different from LA's, and LA's look a lot different from London's. So that's really interesting to kind of get into in more detail. We're studying long-term programs, which at CAS is the Science Action Club, and then also online programs through iNat and Zooniverse. Next slide. And so in these 3 settings, we're collecting these data. So for the field-based programs, we're doing youth observations, where I essentially go to a BioBlitz or a Science Action Club and follow kids around for 20 minutes and write everything they do down. We also do pre and post surveys, and then interviews with facilitators, the designers of the programs, and with focal youth in long-term programs. And for online, we're just looking at user data online, and we have our online education researchers who are getting deep into analysis on that, and then also interviewing a subset of those online users who are willing to talk to us a little bit more about why they're participating and how they're doing it and what they're getting out of it. Next slide. So these are just some things we're just beginning to find. Again, we're just kind of starting to get into the analysis phase of our project. But one of the first questions we wanted to answer was, what do young people actually do at a BioBlitz? They come and we kind of assume that they take part in all of the steps that adults do, but we're finding that that might not actually be the case. So we kind of identified 5 steps towards generating a biological record at a BioBlitz, which is the overarching goal, to produce a record of an organism at a place and time. And so the following 5 steps are kind of what we decided were how to get to that eventual recording space where you're actually submitting a record of an individual organism. And we found that the first 3 steps, exploring, discovering, and then observing and identifying, happen fairly often with youth. Pretty more often than not are they doing all those things and kind of either helping or exploring or maybe sharing their finds with people. And then a little bit less often, they're documenting, which just means maybe they're writing something down in their notebook or they're taking a picture but not submitting it. And then the recording is when they actually take that documentation and they submit it to someone that can use it for a scientific purpose or management goal. And we're hoping that that happens the least. It still happens, but not as much as maybe we had assumed when we started this project. And again, that differs per museum and per setting a lot, which is really interesting to look at. Next slide. And these are some setting features that we have deemed have been really impactful. So how these play into the setting overall and how these affect youth participation. So access to tools has been really important, both how many and what's available, and then the accessibility, mostly age-wise. Access to experts as a resource. So for instance, at our BioBlitz, we often have kind of taxonomic experts and amateur people who are really into spiders or really into birds, and kind of their willingness to share that knowledge with youth and help them along the way. And then adult facilitation is really important, which I think is kind of obvious. Obviously, how you're facilitating youth experience during an event can really affect that youth experience there. And then creating opportunities to share knowledge, skills, results, and findings has been really important, especially for that agency part I was talking about, where youth can use what they've learned to go then change something about their life with the scientific things that they've learned throughout the event. And then access to citizen science, which basically means like how easy is it for them to actually submit data. So that can be a lot about tools or how many people are facilitating their experience, but just general access to participating in the citizen science aspect of it. Next slide. And so here are some preliminary findings from online use in iNaturalist. So our online researchers sampled 115 young people in their first year of our research. We found that the majority of those youth made pretty few observations, 1 to 50. And the most observed taxon was insects, which makes sense. And then the majority of the young people, we're calling them visitors. So they kind of just came once or twice and then really never came back to iNaturalist again. But this chart at the bottom shows the number of observations or identifications on the bottom, and then the number of youth on the left. So you can see that there was most youth didn't make any identifications and made very few observations. And in the middle is like very few youth kind of made that middle portion. And then there were some super users at the end who are a few youth that we know that really, really love iNaturalist and also make IDs, but those are pretty few. So for the most part, we're finding a lot of the young people that use iNat are just visitors, often who come to a BioBlitz and use it for 1 or 2 days maybe, and then might not visit it again very often. So again, these are some preliminary findings that we've discovered so far, but we're really just getting into it. So hopefully I can share more soon, but I'll give it over to Allison, I think.
It's Rebecca for just a little bit, and then back to Allison. So thanks so much, Annie. It'll be great to have you give a seminar when we have more findings. So you can talk about all of the work that you all are doing. So one of the things that, as we've built our program in the Citizen Science Department, that we've realized is that this tool or this building block of the BioBlitz can be used, can be scaled, so we can coordinate and have many BioBlitzes across space or time to help answer specific management or science questions. And so today we'll talk specifically about 2 projects, Snapshot Cal Coast and the City Nature Challenge. But I just wanted to mention California Biodiversity Day, which is represented here by this California Biodiversity Atlas photo. We've been working with the state of California to help them plan Biodiversity Day last year, which was a series of BioBlitzes, coordinated BioBlitzes, and we're working with them for the Biodiversity Week in 2020, which will be the September 7th, probably the beginning of the week of September 7th this year. And so we're helping them plan BioBlitzes across the state. So stay tuned for more information about that. But first I wanted to talk about Snapshot Cal Coast. So next slide. So when Allison and I started our program, we really wanted to make sure we did some work in the tide pools. You know, we both have a connection to intertidal ecology and the animals, the invertebrates especially, that live in the tide pools. And so we started working at Pillar Point, which is just south of the Fitzgerald Marine Reserve near Half Moon Bay. And when we originally started working there, although it's immediately adjacent to a state marine protected area, and was specifically left unprotected by new marine protected area rules, there wasn't a species list for this place. And we were working in partnership with the Greater Farallones National Marine Sanctuary to help understand this place. And so we knew that by engaging volunteers, we would be able to not only build a species list, but to build a species atlas. So over the past about 8 years, we've been able to go from no species list to the data that you see here. Next slide. So we've been able to have a great list of what is found at Pillar Point. And even though we've been there a lot, we're still finding new species once in a while out at Pillar Point. So next slide. One of the things though, by spending so much time in one place and building a cohort of volunteers that work in one place and know it really well, we were able to start noticing changes. So we witnessed of course, the sea star wasting disease and changes in species abundance and range as a result of the warm water blob, like the increase in this Hopkins rose nudibranch. But we were mostly working in one place, right? So we had a crew of volunteers that know one place really well and can see these changes, but we knew that in order to really answer questions about what's happening along our coast, we needed to scale this work up and down California. Next slide. So we worked in partnership with the Resources Legacy Fund Foundation, actually originally, and now with the Ocean Protection Council to design a program called Snapshot Cal Coast. So every year since 2016, over the first couple weeks in June, we've worked with partners up and down the state of California to document the biodiversity of the California coast. Basically, everyone gets out and makes observations of as many species from as many places as they can along the coast. And we're able to use those data to create a snapshot in time of where species are found and look at those data year over year. And Gia will talk a little bit more about the data analysis resulting from Snapshot Cal Coast. Next slide. And the way we can do this is that there is an amazing network of folks that are working in marine protected areas along the California coast through what's called the Marine Protected Area Collaborative Network. And so we are able to build partners through an existing network. And this is just a little smattering of all of the different partners that we work with. And these folks work together to mobilize their local community. And that might mean getting their marine lab out to the coast. It might mean just getting a small group of their volunteers to the coast to make observations, or it might mean holding a big 100-person BioBlitz. And what we do is we provide people the tools and the training and the resources so they can hold BioBlitzes in ways that work for them. And because we are all working toward the same goal, those data can all be used to answer these big questions. Okay, next slide. So really, it just looks like a regular BioBlitz. So next slide. This is just an example of a day where we took a group of folks out to Frenchman's Reef. Next slide. Next slide. And even though it's just about 14 people, when you made 300, just over 300 observations of about 150 species. Next slide. Everyone is doing this in coordination up and down the coast in ways that work for them. Next slide. So we're able to get, again, this picture of the species that are found along our coast over those 2 weeks. Next slide. And over the endeavor has grown a lot since we started in 2016. You can see overall we've made about 71,000 observations. You can see the geographic spread here. Last year we had that looks like a really big increase. We kind of changed the boundaries that we use to make it more consistent for GEO's analysis, but we still had a huge increase. This year, Snapshot Cal Coast is currently still scheduled for June 1st through 16th, but there's a chance we might move it to offset it 6 months into December, depending on how things are looking with COVID-19. So I'm gonna turn it over to Geo to have him talk about the work that he's doing to analyze these data.
You might need to unmute yourself.
Yeah, there we go. I've unmuted myself successfully. That's great. Hi, everyone. My name's Geo. My expertise really lies in the analysis and synthesis of large ecological data sets. And so I joined the team to really, I joined the team when the Ocean Protection Council, which is the California state agency tasked with the protection of marine and coastal areas, had the incredible foresight to see how useful these data coming out of Snapshot Cal Coast and recorded using iNaturalist, how useful these data can be to monitor the California coast and marine protected areas specifically. So what you see here is an animation of just how many data points there are across the coast. And as you see, there's a big spike every year. That is actually the combination of Snapshot Cal Coast and the City Nature Challenge, which we'll find out about later. But so how can we make use of these data to monitor the coast? Next slide, please. Obviously, one of the biggest issues with these data is that they are biased in a number of ways if we want to find out about ecological insights and not so much about people's behavior in recording biodiversity. And so this figure here on the left shows the activity of 3 of the most prolific iNat users along the coast. And as you can see, they all have sort of different patterns of recording, and they're sort of spread across the coast in different ways. They have different foci. And what that generates when you aggregate all of the data together from all the users throughout the coast is this pattern on the map on the right, which is the observation rate in different places across the coast, with the darker red being higher number of observations. And you can see that that varies a lot across space in a way that is not random at all, but actually is biased by many factors, including where people live and how accessible areas are. Next slide, please. So another bias is that we're incredibly fortunate that people are using iNaturalist more and more. But what that means is that if you just simply look at face value at data for any taxon, and these are just the 4 most widespread taxa across the coast, across the intertidal, everything's going up almost exponentially. But this is really iNat activity more than anything ecological going on. Next slide, please. So I just wanted to provide you a quick example of what we might go about teasing out ecological signal from these data that are noisy, but incredibly useful and incredibly rich. And here I'll put a plug for actually a full seminar. I'll be giving a full seminar on this, so you can join back on April 23rd if you're interested in finding out more. So this is Pysasta ochraceus here, the ochre sea star. And as you can see, the number of records for Pysastas are going up over the years. But what we really want to do is not just look at those data face value, but those data with respect to the background rate of observation, if you will, of species that are associated with Pysasta and are also being recorded in similar rates of observation. And so I can go into more detail into this, and we'll go into more detail, but I don't have time right now. But next slide, if you look at the relationship between those 2 rates of increase, what you get is this relationship here, which is a lot closer to the ecological signal we would expect of Pysasta, which unfortunately saw massive declines due to sea star wasting disease, and fortunately is seeing some recovery across the California coast. So next slide, please. To conclude with this kind of crazy animation here, which is way too fast, but it's all I could load on this presentation. We're building all this information into web-based tools that are co-developed by stakeholders on the California coast, so state agency, tribe representatives, fishermen and volunteers, members of the public, that tell us the kind of information they would like to see and the kind of ecological signals they would like to see out of these data. So please join me on April 23rd if you want to find out more. Thank you.
All right, there we go. I've got to go between my 2 screens, unmute myself, and then go back to that one so I can forward. So finally, the last kind of big scale project that we're going to talk about is the City Nature Challenge. We saved it to the last just because it's actually coming up the soonest right now. And just a reminder that the City Nature Challenge overall is organized by us at the Academy and then the Natural History Museum of Los Angeles County, their community science department down there. The City Nature Challenge has had amazing growth, kind of all on its own. We really had no capacity to do any sort of strategic growth with the City Nature Challenge. It's all been about word of mouth, which is pretty great. But going from San Francisco to versus Los Angeles back in 2016 to cities in the US in 2017 to international in 2018, we've had amazing growth, which we're always amazed at. This year for 2020, we had over 230 cities signed up to participate in the City Nature Challenge this year in over 35 countries. That's changed a little bit in recent days, but we still have over 200 cities signed up, which is great. And one of the things that we're really proud about for the City Nature Challenge is not just that we managed to get a whole bunch of people out for 4 days and connect to their local nature, really specifically around urban nature in cities, which we do every year, and we do it really well. Those spikes that you see on that graph of observations per week on iNaturalist is the City Nature Challenge, the big ones in 2017, 2018, and 2019. But one of the things that we really love about the City Nature Challenge is that we're also kind of turning people into observers and to naturalists, that the fact that every year that we reset the baseline of the number of observations coming in each week lets us know that we're obviously motivating a lot of people to get out and participate during those 4 days, but for some of those people, that introduction to the world of slowing down and being curious and really looking for nature all around you and then documenting that nature that introduction through the City Nature Challenge actually keeps them making observations throughout the year. So that's one of the things that we really love about the City Nature Challenge, is it's a nice way to introduce people to this idea of becoming an actual observer and recording data. So the City Nature Challenge, the way it works, like I said, we haven't done any sort of strategic outreach to anybody. It's all been about word of mouth, but it's all based on local organizers, people who either work for an organization that has some alignment with connecting people to nature or documenting biodiversity in that area. They basically sign themselves up and say, yes, I'm willing to take the time to organize my city. Some folks are totally volunteers. Their job has nothing to do with nature and biodiversity, and they do it on their own time, which is really amazing as well. And then Leela, who's the manager of citizen science at the Natural History Museum of LA, and I, much like Snapshot Cal Coast, we take the time to basically train all these folks, to give them the resources and the knowledge and all the information they need to successfully make the City Nature Challenge happen in their city. We start working with organizers. This year, we started working with them in September of 2019 to get them ready to have the City Nature Challenge happen in April. And that includes monthly meetings and developing resources and all sorts of stuff, huge Slack channels, things like that, to make it happen. And in past years, what's really driven the City Nature Challenge, the success of the City Nature Challenge, has been BioBlitzes. I love this kind of collage of photos taken from around the world of people getting out. And because again, part of what we're really trying to do is to build community in these places as well. So people being able to get out and get together and document their local nature. Clearly, though, the BioBlitzes are not really happening this year for the 2020 City Nature Challenge. Some cities are still able to hold events, and it's interesting because some cities, like in China and other places, are kind of coming out now of being locked down and are actually able to start actually having more people socialize. So it's kind of interesting to see where our different cities are in the COVID-19 kind of wave right now. But we're really pivoting the City Nature Challenge this year to be about looking for nature close to you. And one of the main changes that we've made this year is we've decided the City Nature Challenge is no longer a competition this year. That in the past, it's always been about, which city can make the most observations and find the most species, and to, get the most people out there documenting. This year, we're really just going to embrace what can we all do collectively in whatever capacity we can, while still following all of our local government regulations. And so this year, the dates, just if you don't know them, April 24th to 27th, or when we're asking people to make observations. The 28th to the 3rd, we kind of give people a week to make sure they get everything uploaded and to work on identifications. And then our results will be announced on May 4th, but it's going to be the collective results that are announced. What did we do all together this year? So we're really focusing people, kind of the messaging that's going out around the City Nature Challenge right now, are asking people, what are the wild species that you can find even inside your house or in your yard? We're asking people, can you attract some new species to your yard? We're giving some resources about how do you actually attract nature to the areas that you live in. We're asking people what wild nature they can find in their neighborhoods, so looking beyond the cultivated plants, but what are those, wild plants that are growing in between those spaces, or what are the insects that are using those cultivated plants as habitat or food sources? And then for people who are able to still go out to their local parks, asking people, what can they find on a solo walk or a walk with their family this year during the City Nature Challenge. So we know our numbers are going to be pretty reduced this year. We were really excited to break 1 million observations in 4 days this year. We have a feeling it's probably not going to happen anymore. But like I said, we still have over 200 cities that are participating, and we're trying to cultivate still a feeling of community, of everyone, in whatever capacity they can, is still going out and trying to connect to nature during this really difficult time. So we've developed some resources to help folks. We have a resource now about exploring nature in and around your home for the City Nature Challenge. The link to it is up there, but like I said, we have a resource that we'll give to you at the end of this as well. And one of the fun things we decided to do is make a challenge specifically for those of us that work or volunteer or are a student at the Cal Academy. We're making it locationless because we know not everybody is based in the Bay Area. But during those 4 days of the City Nature Challenge, if you are interested in participating, we are going to make a user-based challenge where we'll collect your iNat username and we'll put you into this challenge, and we'll see which of us who work with the Cal Academy or have some connection to the Cal Academy can make the most observations and find the most species. And we're also coming up with some other fun prize categories as well, not beyond just big numbers, but looking for the more interesting observations that people make as well. So like I said, we have links to those things on here, but we'll give you a resource at the end that has these links as well if you're interested in being part of the Cal Academy Challenge. So with that, I'm going to turn it over to Tony.
All right, hey folks, thanks Allison. So this is a graph that is just, we're kind of interested to see if how the COVID-19 pandemic and shelter in place has affected iNaturalist. So Kenichi made this this chart. And I'm just gonna basically steal all of his conclusions from it. But if you look at the dotted lines, the green dotted line is our observations being added last year, and the orange dotted line are the number of sessions. Sessions meaning someone's on the website for about 30 minutes or more. And you can see you can see like a nice dip in the middle of the week with peaks during the weekend. On the gray bars are Sundays. So that's pretty much what you'd expect, right? People are out on the weekend adding observations, and maybe throughout the week, people are actually spending time on the website, adding IDs, making comments, et cetera, et cetera. And the solid lines represent the same things, but for 2020. So until about 3/8, we had 3/8, and maybe up until the Friday 3/14, we had kind of your standard dip during the weekend, growth during the weekend. But you can see on Friday, 3/14, we actually had more observations than we did have sessions. So that could be people using the app more and adding more observations via the app. Could also be people adding some CSV, their old data. And you can see that the week starting 3/16, there's definitely less of a dip throughout the week, and it ramps up before the weekend, which is pretty interesting. And you can see over the weekend, it actually ramps up on the weekend. You can see that like the number of observations blows past the number of sessions. So maybe that's more mobile use. I do know someone uploaded an enormous CSV of their old observations to iNaturalist, which caused a bit of a spike. We recommend not doing that because iNaturalist is really about sharing photos that people can evaluate. But yeah, we've only had a week or two of really, big shelter in place for a lot of iNat users. So it'll be interesting to see how this follows. Oh, never mind. So Kenichi tells me that these are verifiable observations, not the CSV observations. So sorry about that. So that big growth would be probably a lot of mobile users, which is pretty interesting. So we'll kind of keep track of this kind of stuff. You can also go to inaturalist.org/stats to kind of just see our day-to-day stats, which is kind of interesting. Next slide, please. All right, so a lot of people think iNaturalist is mainly about getting outside, and it totally is. But it is also a really nerdy thing. So it also involves a lot of clicking and typing on your computer late at night. And one thing that would really help out the community, which anyone, especially we have a lot of experts here, I'm sure, or at least people who are quite familiar with certain taxa or certain places. If you can help add identifications to other people's observations, that would be really, really awesome. Because especially with new users, you want them to be able to feel like, hey, someone actually saw what I posted and they gave me feedback. That really keeps them kind of going, and making that first, like closing that first loop is really, really important. So what we do have an identify page on our website, and there's a tutorial in the document. There's a link to a tutorial in the document that Allison has. And this is me pretending to be Allison or Rebecca. And what you can do is if you can go to the identify page, look at the top left, you can filter for like taxa and a place. So if I want to look at nudibranchs in California, it'll show me all observations of nudibranchs in California that that still need IDs. And you can use keyboard shortcuts to go through them pretty quickly and either agree or disagree, add a comment, and really help out a lot of people. It's pretty fun, actually. It can be kind of addictive. Sometimes it can be frustrating, but sometimes it's usually pretty awesome. So that's one way you could really help out. And next slide, please. While you're identifying, I just want to go over some etiquette. For one thing, you don't you don't need to add an ID. You can feel free to skip something. If you're not sure what something is, if you don't want to weigh in, it's fine. Don't feel obligated to do it. If you do add an ID, please add an ID that you can independently verify. So don't just agree with something, especially, you know, if you're not sure, you know, maybe it is that flower, but maybe there are like 5 other flowers just like it, and you don't know exactly which one it's not. So really add an ID if you can only independently verify it. You also don't have to feel obligated to identify the species level. It's fine to go to genus or family or whatever. Anything kind of helps to refine it. You know, there's a lot of people out there who don't know the difference between a beetle and a bug. So even adding that idea will help them out and teach them something. It's good to remember that, especially right now, this is the first time that a lot of people are really interacting with the naturalist community. So it's really important to be helpful and encouraging. Maybe they put up a blurry photograph. Maybe they didn't photograph the right part of the plant or the animal to help you ID it. It's best to just kind of give them some advice. Say, hey, like this is, you know, next time, you know, it'd be great to have some photos of the leaves of the tree, not just one from, you know, 20 feet away. But say it nicer than that. Just be encouraging and helpful. You know, it's a great way to kind of turn people into really good naturalists. Remember that it's fine to disagree with people. If you do, please be civil, and please cite evidence. Even if you're an expert and you know what something is, you should be making an argument citing evidence. And if you have any issues, any like interpersonal issues, you can email us at help@iNaturalist.org. And I just wanted to put an example here. Kenichi does a really good job of IDing and actually adding helpful comments. So here's just an example of you know, him linking to resources that someone could actually look at and telling someone, hey, next time try to photograph the leaves and the fruit. That would really help us identify it. So that kind of stuff is really what iNaturalist is for. It's for us teaching each other about how to become better naturalists. Next slide, please. So another thing you can do if you're stuck at home is, especially if you have a backlog of photos, is to add them to iNaturalist. One of our top users, Greg Lasley, he added photos all the way dating back to the 60s, which is pretty amazing. And right now we've got a few people. One gentleman wrote in, he is adding like thousands of observations. And he wrote, just starting to enter 2010, I need to get all the way to 2018. And this man from Ecuador, Frank Dietza, says he's going to upload all his photos of 16 years in Ecuador. And that's just amazing, amazing data to have, especially longitudinal data. And if these photos are on your computer, you can use our web uploader, which I have a picture of here at the bottom. So you can upload maybe about 50 photos at a time. You can combine them, and you can kind of edit their location, their date, and add an ID to them. And it's great stuff to have up on iNaturalist. Next slide, please. I just want to point out that, you know, maybe if the photo is from a long time ago and you didn't keep, you know, really good records, when you add a location to the observation, you'll see a red circle appear when you click on the map. You can then resize that circle to be bigger or smaller. So just make sure that circle covers where you believe you saw the organism, so that other people know exactly how precise or imprecise you are. That's really important for old observations, just so there's transparency as to how accurate that location is. Next slide. And I just want to say that this is a meme that was made by someone on the iNaturalist Discord server. A lot of people upload observations with no ID. And so even adding an ID of like plants will help it be found by maybe someone who can identify plants. You know, if there's an expert in plants, maybe you aren't, but even by saying plants, you're helping that be seen and you're giving feedback to that person. And so, you know, like the meme says, it ain't much, but it's honest work. So anything you do will be a big contribution to iNaturalist. And you'll find yourself learning a lot and probably wanting to explore a lot more once you can. And I think that's about it.
All right, so I just have a couple things. Allison and I have been talking about challenges around the things that Tony just described, that maybe we could put these challenges to all of you. And one of them is to look at the Bay Area observations. So these are all the observations that have been made in the San Francisco Bay Area. This is the area that we use for the City Nature Challenge. So next slide, Allison. So the top number is all of the observations, and this bottom set of numbers that just appeared are those that are research grade. So next slide. It's about 62% of the observations are research grade, and while we're home, all of us can contribute to this and try to improve that ratio. We can't necessarily look at just the straight numbers because people are adding observations, but I think we can increase that percentage, and that's a nice challenge for us to just look at the Bay Area and use the tools like Tony described and try to get the things that we know from here to research grade. Next slide. The other thing that you can do is you can help us identify Snapshot Cal Coast observations in the same way. The more of the observations that are research grade for Snapshot Cal Coast, it makes Gio's job easier because he's using verified data and has more of it. And also one of the things that Gio's looking at is rare or really, really unique taxa. And sometimes along the coast, that means it's something dead, like a shell or seaweed. And if we can, there's a way to, next slide, Allison. You can mark things dead or alive, and for shells, this would really help us differentiate between live snails, for example, that are really rare, or snail shells, which can also tell us something, but it will help us differentiate between those things. So it'd be great to have your help with those two kind of endeavors. Next slide. So we also wanted to highlight a few things that you can do that are not iNaturalist and not our work. And we wanted to especially highlight the Zooniverse, which is an amazing platform and has a huge suite of projects that you can go to and basically help either identify things from pictures or transcribe things like ship logs to look at past climate data, or literally almost every single thing you could think of. There's something for everybody on the Zooniverse. So I encourage you to check it out and find something that kind of strikes your fancy. Next slide. I wanted to especially highlight one of the projects that's a California wildflower or flower phenology project. Next slide. Where there are photos or scans from California herbaria. Next slide. And the ask is really to help transcribe these labels. And so many of you are familiar with this because we've participated in CalBug and other notes from nature projects, but this is a really good example because it's from California. And as we're all thinking about things that we might want to do with our data, these are some good examples. So not only you can do the work, and you can also get inspiration for the resources that we have that we might want to put on a platform on Zooniverse. Okay, next slide. There's also some other stuff that's really fun. Like this is a burrowing owl webcam, which is really, you can just look at the webcam and tag whether or not you see a burrowing owl or what species you see in these pictures. So it's fun, and sometimes you see nothing, but sometimes you see really cool things, and it's really helpful to the folks that have those data. Next slide. So there is a way to build projects on Zooniverse. I'm not going to go through it now, but I have on the resources that we'll share, there is a link to put your name to sign up to be involved in conversations about how the Academy can start one or more Zooniverse projects, especially for our volunteers that no longer can come into the Academy. We can set up projects that are just targeted for a specific set of people, not public. And so there are lots of great things we can do together using the Zooniverse platform. So look for that link. All right, I think that's it, and I'm turning it over to Allison.
All right, to end off, we just wanted to point to another cool project that you can do from home right now, and especially right now where there's not nearly as many lights out at night these days. The Globe at Night is another really cool project, and they have some really great, they have a dark sky week in April and another big day in May that they're looking for people to make observations, and it's simple as going out into your backyard and basically looking to see what constellations you can see and which are still obscured. So that's another cool project to check out. And finally, I just wanted to point out there's some really amazing citizen science and open innovation going on right now around the coronavirus and COVID-19. So if you're interested specifically in helping out with those projects, Fold It, which is like an online protein folding game that people participate in, has been doing some really amazing work around COVID-19 and getting their users to kind of focus on work around that right now. So that's a cool resource you can go check out. There's another platform called Eterna, which is about RNA that's also working on specifically kind of vaccine stuff around COVID-19, which I recommend checking out if you're interested in participating in that way. There's also other really cool kind of like hackathon and crowdsourcing work that's going on around COVID-19. There's a pandemic response hackathon that actually starts tomorrow, starts on the 27th and goes for a few days to get basically people who work in technology and people who work on front lines of medicine together to kind of tackle and track the pandemic. And then there's this amazing resource that the USGS has put together around COVID-19 open innovation efforts. So, and it's way longer than just this page. I couldn't actually capture the whole page. It's huge around things that they're putting together to just kind of help people connect to things that you can do around the pandemic right now. So with that, this link, this bit.ly up here, bit.ly/SitSciLinks is the link that we have that has all of the resources of everything that we've talked about put together, all the links, the Cal Academy Challenge, all the identify tools on iNat, all that sort of stuff if you're interested in checking them out. And thanks so much for listening to Citizen Science Now. Yes, I'm going to put the link in the chat right now.
I just threw it in there. Okay, so well, I guess it's hard to give you guys a round of applause, but everybody can join me in a virtual but probably mostly silent round of applause for our citizen science team. Woohoo! That was awesome. Thank you guys so much. We do have a few minutes for questions. If anybody has any questions, just jump in to the chat and type a question mark, and I'll call on you to unmute yourself and ask your question. There was a conversation that was happening during the talk between Kenichi and Gio that I don't know if anybody had a chance to see the discussion they were having, but I wanted to give them a chance to talk about it out loud. Instead of in virtual. So if Gio or Kenichi wanna reiterate what you guys were discussing, please unmute yourself.
Yeah, so Kenichi was asking basically what the Paysaster associated species curve actually showed from the graph. So if you remember, I was showing the observations of Paysaster and then the observations of species associated with Paysaster, and that can be interpreted in a number of ways and indeed determined in a number of ways, and we've thought about it in many different ways. But what that was is basically based on a clustering of all observations across the coast. It's actually the set of species that is most often being observed together with Paysaster in the same place at the same time and by the same users, with place being defined as 2 kilometer or so grid square. So that's what that curve is showing. It's an estimate of the observation frequency that is relevant to Paysaster and species around it.
Great, thanks. Natalie, I think has a question. You want to go ahead and unmute yourself, Natalie?
Yeah, so thank you, team. That was amazing. So Annie talked about the biases in terms of the young people and they prefer to record insects, but in general, what do you find that people sort of gravitate to, and how does that affect your sort of data that you produce?
Yeah, I mean, I think, and Allison and Rebecca know this better than I do, but I think that's pretty a common trend for all users, not just youth, especially for beginner users, to more often observe insects. And it makes sense when you think about it. When you go to a BioBlitz or you go outside, they're kind of the most ubiquitous things to find and the easiest things to find. And sorry, what was the second part of your question?
So how does that affect the sort of resultant data and the things that you're starting to analyze?
It really doesn't affect it too much. We're not really focused on what youth are observing specifically. We're more interested in what they're participating in citizen science overall, not necessarily what they're submitting, if that makes sense. So the online team of researchers that we have at Open University are looking in a little bit more detail about what's being submitted to iNaturalist and are getting more deep into those kinds of things. But for my part of the research, which is mostly the field-based aspect, it's not a huge issue that they are more interested in insects versus plants or birds. We're just kind of interested that they're observing things at all.
And I think overall, you know, when you think about people making observations, a lot of people are using their phones, right? So it's things that are easy to take photos of with your phone. So lots of plants get taken to get lots of pictures of, you know, insects that are not super fast and running away from you. It's a lot harder to get bird photos with your phone, stuff like that. And so I know, at least in the work that Gio does, you know, he's kind of looking at those biases and basically, you know, understanding that if there's not enough observations of a particular thing, that it's not actually included in those analyses as well. Luckily, in the intertidal, a lot of stuff just stands still. Nothing moves too fast in the intertidal except for the fish. But who cares about things with backbones anyways? So.
Yeah, sorry, if I can just add a thing. In my experience, actually, the biases are very similar to many other ecological data sources. So, you know, taking a picture of what's easy to take a picture of is just a detection issue. That's exactly the same as being able to hear some bird songs and not others, or being able to spot some birds or not others. So, and then, you know, the spatial and temporal biases are, you know, maybe more severe than other data sources, but they're basically the same. So the tools are out there to figure out those biases. And yeah, so.
I saw there was a question in the chat from Anna, who was asking, can we create a project to enlist the help of a global community to include observations of a specific genus? And I mean, the answer is yes. You could create a project that helps aggregate a bunch of different observations, but what you're really talking about is a campaign. Like, you don't really need a project to find all of the observations of one genus, because you can just search for that in iNat. You can just type the genus in the top bar and see who's made observations. But what it sounds like you would want to do is to encourage people to make those observations, and you can do that via iNaturalist by encouraging people that have already made observations of that genus, encouraging them to make more, or you could do a social media campaign asking people to make observations. So there are reasons to make a project. If you wanted to have a URL to really like communicate with people, you can do that, but you don't have to. Alice and I are happy to talk to you about the differences offline, or what in our experience has worked and would help.
Okay, I think unless there's any other questions, we'll thank these guys again. And I know I'm certainly ready for some iNaturalist observations in my self-imposed quarantine.
Yes, everybody sign up for the challenge. Go to that resource link and get yourself onto that Google form so we know your iNat name and we can add you to our challenge for the City Nature Challenge.
And again, the link is in the chat. I'll repost it again down at the bottom one last time if anybody's looking for it. Thank you, guys.
Thanks so much, everybody.
Thank you.
Everybody.
Bye.
Thanks so much, everyone.
Bye.
That's fantastic. Thank you.
I
Thanks,
think
guys.
I'm recording now, Natalie. Maybe I can do it.
Okay, I'll turn it off.
I stopped it.